thanks everyone for coming to this webinar. So Ben Hibbs founded Klein Australia in 2020, which probably was a hard year to start. He looks after Nikon General Microscopy and distributes other things such as clearing reagents and sample holders, and does training around light microscopy space. So he had a background in biochemistry, and he was an image facility manager at University of Melbourne for a couple of years before he went to Coherent Scientific. So now he's made a progression through to distributing, and he's going to talk to us about SunJin Lab optical clearing products.
Yeah, that's right. Thanks so much, Deb. So can everyone see the PowerPoint slide okay, or is that not sharing properly?
Looking good.
It's looking good? Great, you can see it? Yes, you're never quite sure which window I'm sharing sometimes. Fantastic. So, so my name's Ben. I just wanted to run through the clearing reagents available from SunJin Labs, but we'll delve into some other clearing methods and why you might clear, what type of samples clear, and how clearing reagents work. So we'll dive in, and if you have any questions, jump in or put it in the chat and we can look through it later. So I'd really like to just quickly run through, the company background, but we'll really focus on tissue clearing and why you would clear, some of the backgrounds and processes, and some of the various clearing techniques, because there's a lot of different techniques out there. And then we'll look at what's available from SunJin Labs and the specific applications that we can call upon there. So SunJin Labs have been around for quite a while, and they're really focused only on optical clearing techniques and reagents. And their key product is the RapidClear clearing reagent. And they also produce a few other products that we'll quickly step through. And in Australia, they're distributed by me through my company, Klein Australia. So we've got our links down there, kleinaustralia.com.au and sunjinlab.com. So RapidClear are their main products, and we'll run through what those are. Basically, we have different refractive indexes depending on the type of samples that you're clearing and what type of refractive index that you need to achieve in your sample. We'll discuss why that's important. And we have a few other clearing reagents to use with other clearing methods as a final refractive index matching step. Some other products that SunJin Labs produce are what they call eye spacers, which are small adhesive stickers and wells to attach to slides. They're basically a way as like a rectangular or circular sample holder to contain your tissue section in place and hold the RapidClear as a mounting medium in place. So there's lots of different thicknesses. You can stack them to build up the thickness that you need for your sample. They're much more flexible for clearing than a multi-well chambered slide or something like that, because you minimize the amount of clearing reagent that you use, and you can seal it with a cover slip for imaging or storage. And they've got a whole range of these products. And we've got some samples, by the way, with Deb. So if any of you want to try these out, just contact Deb. And the final product that SunJin Labs have is the flow tissue prepared slides. So these are prepared tissue sections. There's 3 different types of tissues available: kidney, brain, and another one that are mounted in RapidClear 1.52. So these are 550 micron thick sections with 3 colors for instrument testing, imaging, and checking systems. They're like a thicker cleared alternative to the Life Technologies prepared slides. 3 different sample types. Okay, so why would you clear? When you're imaging on an optical microscope, you're limited in how far you can image and the resolution that you can achieve through a variety of issues. But as you image deeper into a sample, the scatter caused by refractive index differences and opaque structures becomes more and more of an issue. So these all limit the resolution and the imaging depth that you can achieve in optical microscopy. And various techniques have different ways to try and address this, but they're all limited in some way by the physics of the scatter and the behavior of light as it travels through your sample. So if you have an uncleared embryo, obviously it's scattering a lot of light, we can't see through it, and the cleared embryo in the same dimensions after it's been through the clearing process is almost optically transparent. And then finally, if that's labeled with an antibody, we can image that in much higher resolution throughout the entire sample because it's a cleared sample. So there are many different examples with the rapid clear reagents. They have a lot of experience with various samples. So if you have something unusual that you'd like to try, there's usually a data sheet or something to look at on their website, or we can direct you to a protocol that has worked before on different types of samples, even including bone, insects, plants, which are all have been used with great success. Okay, so I want to briefly run through why you might consider clearing. And I guess, you know, if you're in this webinar, you're interested in clearing, but it's worth just having a think about what clearing might do as an extra step to help with the results of your imaging. So if we look at a thin section, we can look at structures in a 2D view. Maybe we can make that section a little bit thicker to build up a 3D view. But if we want to image the structures in 3D over an organ, we really need to maybe sequentially section an image to build up our entire organ view and allow us to image that in high resolution that way. But that's a lot of time-consuming steps. So our other approach could be to clear the entire organ and image it all at once in high resolution using the clearing reagents. So the question, of course, is how can we make tissues transparent? But even more importantly, why can't we see through tissues in the first place? You know, what's blocking our view? If you look at some sort of organ, you know, it's light's not getting through it. So what process is that? If we just consider a model, say of milk, which is suspended fat and protein droplets in water, it looks white. And if we consider how light is traveling through the substance, through milk, it's having a lot of scatter. So the light is traveling in a lot of different directions and being moved in all directions. Whereas when it travels in air or one consistent medium that's transparent, the light is not being scattered. So the refractive index of the different oil droplets and the different substances in milk is what's causing the different scatters, the light to scatter in different directions. Now, of course, if we could match the refractive index of the water, of the media around those droplets to bring the refractive index up to that level of those droplets, we would scatter less as we transition through the mounting media into the droplets and out of the droplets. And if we can reduce the scatter, then we can increase our transparency. Matching the refractive index causes less scatter. Now, the refractive index refers to how much slower light propagates through the substance when compared to a vacuum. So all transparent materials have a refractive index. For air, for example, of 1, for water of 1.3, passing through materials of different refractive indices causes scatter, and when that happens many times, a lack of transparency in your material. Now, of course, it's not just differences in refractive index which cause scatter in your materials. There's a whole range of pigments of different materials and things in biological samples which can limit optical imaging. In animals, for example, there's melanin, hemoglobin, they're all compounds that absorb light and are pigments. In plants, the whole purpose of, you know, many plant cells is to collect and utilize light, chlorophylls and other compounds cause scatter and pigments to absorption of light. And these are unaffected by clearing techniques. They're not a refractive index situation. So if you want to image deeply into these sort of samples or tissues which have these pigments, they require decolorization, diffusion to remove the pigment, or genetic modification with a pigment-free albino phenotype. Another issue is autofluorescence of background signal in the sample, and that's not necessarily removed by clearing methods. Techniques to combat the autofluorescence really depends on what the autofluorescence cause is. And it's still the case that if you're doing thin, small sections that have autofluorescence, then, moving into a cleared, thicker section will probably not help with the autofluorescence issues. And then, of course, the microscopes themselves have limitations and limits on how deep and in what sort of ways they can image, and things like the working distance of the objective, the shorter wavelengths of light scatter more as compared to longer wavelengths of light, and that's why imaging into the red and the near IR can penetrate much more deeply, and that's the one of the benefits of multiphoton systems. Of course, specialized instrumentation can help get around some of these issues, such as multiphoton, the light sheet systems. But if we can match our refractive index, we can enhance those techniques as well. So let's have a really quick look at a little idea of what matching a refractive index means. So we have some glass marbles in a jar. We can see the marbles, but as soon as we put RapidClear in with the same refractive index as the marbles, they almost entirely disappear, and we are clearing the sample. And for something a little bit more biologically relevant, we play this little demonstration. If we go back to the old example of milk, and it's a substance with suspended droplets, so they're highly scattering, the milk appears white due to all that scatter, and we put in water in one sample to the same amount of the equivalent of the RapidClear that we're going to put into the first sample. Now, RapidClear is a little bit more viscous, so we do have to go through a mixing step with the milk, and once that's mixed properly into the sample, we will see the refractive index has been matched. The oil droplets have not been removed, they're still present in the sample, but the sample is now optically transparent. And we'll just run through the viscosity and why that's important in samples in a moment as well. Okay, so then what do those results mean? On the image on the left, if I just go back to the start here, that sample is in PBS, and on the right, it's in RapidClear, and this is a 500 micron mouse brain slice. So if we start moving through the sample, we've already disappeared. We've already gone as deep as we can in the PBS sample, whereas the RapidClear sample, we can image and resolve structures throughout the entire 500 micron thick mouse brain slice. So in this case, the limitation to the depth of imaging was due to the scatter of the sample, and clearing with the RapidClear reagent allowed us to image deeply into the sample. So if we can make our substance scatter-free, then that's tissue clearing, and that will allow us to do deeper 3D imaging. So there are many different clearing methods, and if you've come across tissue clearing in the past, you'll be familiar with some of these methods. So I want to run through a little bit of the methods that these different clearing reagents use, and run through how RapidClear fits in with all of that. And of course, some sample types are very unique and require very specific clearing protocols. Okay, so broadly speaking, we could split up tissue clearing methods into two broad areas: solvent-based methods and aqueous-based methods. And RapidClear is an aqueous-based method. Solvent-based methods, if we have a quick look at those first. Once we click through. Fantastic. So solvent-based, and we'll just talk briefly about a couple here, BAB and DISCO. Broadly speaking, we have a two-step process where we dehydrate and delipidate with a solvent process, and then apply a clearing solvent and match the refractive index as step 2. So The key advantages of these is that they're often compatible with immunostaining. They can be very efficient and relatively cheap to process. The disadvantages that we can come across with some of these techniques, the solvents can affect the fluorescent probes that you have mounted in your sample. They can cause autofluorescence themselves as part of their process. The very the strong nature of the delipidating, removing the fats, can cause tissue shrinkage. It can actually dissolve the glues through the solvent that's used in the mounting media, dissolve the glues in the cement in the objectives. So these techniques, you want to be really sure that, check with the microscopy facility staff before you start down the path of using these techniques to check they're compatible with the microscope. And of course, these systems can be toxic as well. And if you're studying lipids, not the technique to be using. If we look at some of the aqueous-based methods now, we can look at three sort of broad areas underneath the aqueous-based methods. And the initial one, the hydrogel methods. These methods are really about a three-step process of mounting and immobilizing the organ, or the section in a hydrogel to provide it with structure and support. Step 3 is a delipidating method and with a detergent solution. And step 3, using an aqueous clearing method to match the refractive index of the mounting media to the sample. So as a final step, this mount in a refractive index matching optical clearing agent can be a clearing agent like RapidClear. So we have specific products designed for clarity, for example, as a hydrogel mounted process. And this can be really useful for whole organ mounts for very delicate large samples, which require more support as they're being imaged. And just as an example, clarity combined with other clearing techniques, so combined with RapidClear. So before clarity in the brain organ, after clarity, we can see that it has been optically cleared to a certain extent, but as a final step, clearing with an optical clearing, an aqueous clearing reagent such as RapidClear produces an optically transparent sample. And then we have the immunostaining, the fluorescence image. Okay, so the advantages of these systems is that they're compatible, or some of these systems can be compatible with fluorescent proteins. It can conserve very delicate samples and not change the size and structure of the organ. The complex sort of protocol can mean that these can be quite a time-consuming and complicated process. Some of the reagents are toxic, and they are still a delipidating method. So we just wanted to talk briefly about disco and clarity as two of these techniques. If we just can briefly look at the structure of some of these, the techniques can really differ on what fluorophores they're compatible with. So it's really important to make sure that you're picking a clearing method that's compatible with your samples and your fluorophores that you're using. And the main thing is that the imaging medium that you're mounting your sample in for imaging can be toxic for some of the techniques such as disco. Okay, so if we look at the one-step clearing methods under the aqueous-based headline, we have RapidClear and a whole range of other systems as well. So these are all techniques designed to match the refractive index of a tissue. And this is what I discussed at the beginning, that if you match the refractive index of a tissue, you reduce the scatter in the sample. So in an aqueous clearing solution, we are raising the refractive index of our buffer of our water to match whatever the refractive index is of the sample. And different samples might have different refractive indices as well. So we can if we have the flexibility to fine tune our clearing reagent, then we can produce the best results. We've just got some examples of different materials. So sucrose, for example, has a refractive index at 60% of concentration in water. That has a refractive index of 1.44. So even a sugar in water produces a high refractive index liquid. Glycerol is another compound with high refractive index. Fructose, TDE is a compound that, a material that can get a high refractive index as well. Now, all of these are techniques that can be used as clearing reagents, but how well the clearing reagent penetrates into the tissue and clears the entire tissue is also important. Now, most of these are compatible with a very wide range of fluorophores. They don't remove the lipids as long as you're not doing the clarity or the lipid removing step. They usually maintain the volume and they're quite easy to use as a one-step process. And we'll run through what that step is. And of course, I'm talking about lots of different variations on protocols under this. So there's not necessarily all of these are equally good at conserving fluorescent probes and lipids and the structures as others. Most of these one-step clearing solutions are not particularly good at clearing quite deep samples though. And often when you add, when you add sugar to water, for example, you have a high percentage sucrose solution, it's a very viscous liquid. With a high viscosity liquid, trying to mount your sample in that can cause maybe precipitation, crystals in your sample or air bubbles, and that will dramatically affect your imaging and obviously cause scatter in the sample. So if we match the refractive index of the sample we just have a bit of a table of comparison of a few of these clearing techniques. And depending on what sort of adjuncts you use with these samples, it will have different effects on the ability of these materials to clear samples. But broadly speaking, most of these methods are suited to relatively thin sections of 200 to 300 microns. Most of them are compatible with a wide range of fluorescent proteins, but some materials can precipitate in storage or change color, and some have a narrow range of refractive index, which is available. And SunJin Labs would like to point out that most have a narrow range of optimal clearing range. If we look at RapidClear, so SunJin Labs developed RapidClear as a method to improve upon some of these one-step clearing solutions. So RapidClear comes in the 3 different forms of 3 different refractive indexes, 1.47 to 1.52. And due to the materials in RapidClear, the penetration of the clearing reagent is much better. So you can see some of the examples that we've been showing are much thicker sections. The whole organ labeling clearing, for example, though, is used in conjunction with clarity. So we just want to point out that if you are stepping up to a whole entire organ, that you need another step to really clear the sample. But RapidClear as a one-step solution with a prepared section produces very good results across much thicker sections than other one-step clearing methods. And it's also low toxicity with low viscosity, and you can store it for a long time. Okay, so just some examples of different organs. There's a lot of research. A lot of researchers using RapidClear on a very wide range of samples. So even if you've got something really odd, chances are someone has some experience with it to help us inform how to apply it. And the point is RapidClear is very consistent across a wide range of wavelengths, and it provides clearing and a high refractive index across all of the visible wavelengths and into the near IR for microscopy imaging. And yes, so the refractive index of 1.52 is the same as the refractive index of glass. Okay, so the process of OneStep clearing, I'll just briefly run through some of the, broadly speaking, some of the steps of using something like RapidClear. So after you have fixed and permeabilized and immunostained your sample, you can apply RapidClear or apply staining later. Staining is compatible with RapidClear and some other clearing reagents, but because they're quite viscous mediums compared to just a straight buffer, the staining is not as good as it is in buffer. So prepare all of your staining, fixation, and permeabilization before you apply the clearing reagent. So for the SunJin Labs, you have your slide. The ice spacer is put on top of the slide and you put your sample into the ice spacer. But of course, it's compatible with any method to clear your sample. And if you improve the light scattering, you improve your final imaging resolution. So I wanted to briefly talk about some of the considerations for how you might approach imaging with a cleared sample and what a cleared sample might mean in terms of picking a microscope to choose from. So just as a brief mention, for a very high resolution objective, so 60 times objective with a high NA will be an oil immersion objective. The oil immersion has a refractive index of 1.5 or close to that. The glass of your cover slip has a refractive index of 1.52. And if you're imaging into something with a refractive index of water, so these beads being imaged in water with a refractive index of 1.3, we have spherical aberration caused by the difference in refractive index. And this causes the PSF, the point spread function of the bead, to be elongated in Z, which lowers our consequent resolution. If the same sample is mounted the same distance from the objective in RapidClear, the refractive index matching means that we have much less spherical aberration. We get higher signal at the point of the bead, and the structure is much truer to the actual physical structure that's being imaged by the microscope. And just as an example, for a 300 micron thick sample to zoom through something, if we have something that is consistently showing the same refractive index throughout the sample, we can achieve an excellent resolution throughout the entire sample. Whereas if we had imaged this without a clearing reagent, this spherical aberration will cause our resolution to decrease as we proceed deeper and deeper into the sample. Okay, now really briefly, I just wanted to talk about the last aqueous-based method termed hyperhydration. And we've probably sort of covered a couple of these grounds as well. This is really a method around delipidating and then hyperhydrating using a urea method. So this method is often quite time consuming in terms of the time it requires to be incubated with the reagents, and it's not compatible with all immunostaining steps in some cases as well. And due to the hyperhydration process, it can actually expand the tissue, which can be beneficial in tissue expansion microscopy. But if you're imaging structural information and comparing it to other techniques, that's not so useful. So I wanted to run through some practical suggestions in tissue clearing, not just for RapidClear, but for other clearing techniques as well. So really broadly speaking, if we look at different levels of structures and different types of samples, if we have an entire organ or whole animal or a large physical structures, having a two-step process or a solvent and delipidization process followed by optical clearing will produce the best results with those types of difficult samples. And that's where a light sheet microscope, which can image over a much larger volume than a conventional confocal or inverted or wide field microscope, where they can really come into play. These systems will require a long working distance objective to penetrate into the large structures. Where we really see the biggest benefit for the one-step clearing methods like Rapid Clear and the other methods available is around the thick tissue sections, organoids, insect tissues, plant tissues, where you're not trying to clear an entire animal, but you're trying to improve your resolution over a thick section to gain greater understanding of the context of your structures and proteins of interest. And these systems are best suited to instruments like confocal microscopes, inverted confocal microscopes, where you can simply use your existing protocols and microscopes, clear the samples, and then by matching to an immersion objective and cleared sample, you can gain much better results across the sample. Okay, so if we just to look at a few examples. So this is the light sheet imaging first, and then I think, oh, no, we're slowed down. So basically, it depends on what type of system that you have access to, of course, but often quite a lot of the time, imaging into a section is limited by the scatter of the section on an inverted microscope, not the physical constraints of the microscope. Okay, have we got this one? Right, and then the imaging on the confocal mode, we were able to image as compared to the light sheet microscope in high resolution with this sample. So this is comparing the two types of modalities. The conventional inverted confocal was able to achieve a higher resolution, but less area. The light sheet systems can capture a very large area very efficiently. And if we just look at this 3D structure, on the left, we are going up and down in Z, and on the right, the entire structure is being displayed in 3D. So by clearing, we're able to image all of these fine details on both sides of the structure. Okay, so just some more information around imaging tips. If you can match the refractive index, it means that inverted microscopes are suitable. So the systems used for routine imaging like confocals, the spinning disc confocal microscopes at IMB are all suitable for cleared sections. For large samples, light sheet based systems can be really well suited. And when I'm talking about light sheet systems, we're talking about more larger scale systems, not something like the lattice light sheet, which is designed for high resolution, high speed imaging of live cells. The imaging depth is always dependent on the refractive index of the sample and how well it can be matched to your objective and mounting materials, to the working distance of the objective, to how well the sample can be cleared. And we include the pigments and other scattering compounds in that as well. And of course, the stability of your fluorophores, because if you're imaging a very thick section, the photo stability becomes really important as you image through that. And just to discuss, if you're looking at using the inverted confocal microscopes, it's really dependent on the installed objectives. So things like the working distance, whether the objective has a correction collar, the magnification that's available, and the immersion medium that the objective has are all really important. Just to discuss that in a little bit more detail, the resolution is dependent on the numerical aperture. And if the objective has a correction collar, that means that you can compensate for differences in refractive index and optimize your essentially your spherical aberration in your sample. And there are many different immersion objectives available. You probably have a few at the facility. Just some examples of different refractive index from water 1.3 to glycerol at 1.44. And of course, the oil objectives 1.515 or 1.52 for high resolution imaging. So very generally for cleared samples, if you have some sort of immersion objective, it will be more suitable than an air objective for imaging cleared sections. Air objectives will have significant aberrations imaging into a cleared section. And if you can have an alternative, that will produce better results. Just as an example of a Nikon objective for a clearing optimized objective, other manufacturers, of course, have other objectives available aimed at clearing on inverted microscopes as well. An objective like this is suitable for both upright and inverted configurations, and it has a working distance of 5.5 in the upright configuration and a very wide correctable range. So other manufacturers have similar sort of objectives in the same sort of realm. So with that long working distance and that large correction range, we can image over a very thick section. So with a 5mm mouse brain section, this is imaging over 5mm in an upright configuration, not an inverted configuration in this method. And using this rapid clear sample, we're still gaining useful signal and information even at 3.5, 4mm and beyond. So that's the sort of steps that the clearing can help with. And then you just need an objective and microscope that can accommodate that as well. So broadly speaking, we talked about clearing in organs and clearing in sections. If we talk really briefly about clearing in sections, we can look at that in quite a simple process of perfusion, fixation, permeabilization, and immunostaining as your conventional steps, and then rapid clear as the last step before, well, mounting that and doing that on the slide and as the last step before confocal imaging. And as an example, even imaging through bone into bone marrow into a mouse femur is also possible. If we have a look in here, we can get down to the single cell level as a whole organ, which gives us a lot of context in the wider organ perspective. And individual cell nuclei are labeled in green. Okay, now if we talk about a larger scale organ, so imaging a whole mouse brain, this is a GFP labeled sample. We talked about using clarity or another step in conjunction with rapid clear. And in this step, we've used rapid clear at 1.47, so a lower refractive index than what we can achieve with the thinner cleared sections. And this means that we can get better penetration through the entire sample and not try and raise the entire sample to 1.52. It just gives us a little bit better results through the entire large organ. But this step is a bit more complex because we've done clarity as a method on this entire organ before we've used rapid clear to match the refractive index of the sample. So the key steps in this are mounting in a hydrogel and using a detergent to delipidate and remove lipids from the sample. Immunostaining is run after that, and then rapid clear is the last step before imaging to raise the refractive index in the entire sample and get those excellent results through the entire volume of the sample. And just as another example, imaging the brain using cleared sections gets very good results across the entire section and organ. And we have a specific product for compatibility with clarity, the Rapid Clear CS for mounting and clearing entire organs. Okay, so just as a bit of a summary, we've journeyed through quite a few different clearing techniques and some of the theory. Briefly speaking, clearing is a way to increase resolution and increase depth and the ability to image through an entire or thicker section of your sample. This means that you can understand the context of your structures in much more detail. There are many different techniques, and some are very sample dependent. So if you have an unusual sample, there's certainly a technique out there, probably a technique out there, or maybe there's one to be waiting to be developed. And Rapid Clear is a simple process for clearing for a wide variety of samples. So please get in touch with me if you ever want to try something out. You have some samples with Deborah to try. So please let me know how you go, and contact me for quotes and more information.
Great. Thanks so much, Ben. That was a really great
My pleasure.
presentation. Lots of good examples in there.